Okay, so I hope for the few more people who come and who will come and join us, we'll change the name. So if I can give you again the link for today's attendance, if you want to click, look at the chat box function. I hope you get a good internet connection. Okay, so you can just click on the I attend, but if you fail to do that, please tell your section leader to rectify the issues or give the list of the names to me. Okay, so tonight is the viral pathogen and associated diseases. What you need to learn tonight is you need to know how many types of viruses, that is commonly associated with human diseases. And along the way, if you got any questions, if you're not happy, if you are confused, please raise up your hand and you can ask anytime. So the objective for today's session, you will need to describe the general properties of the virus. You need to explain replication. Replication means they're doubling, okay? They become double because they need to survive like human being. So we get married and we got kids. I mean, this is to sustain our life, or else we will stop, okay? We will die and no further descendant. So in the case of the virus, as long as it's considered as an organism, they have to replicate themselves. And this is true for both RNA and DNA viruses. But what is really the RNA and DNA viruses? So what really the differences? So that's the thing that we need to learn tonight. Also, the most important thing is how does it make sense to your topic or to your career or to your course, because you are the future professional nurse. You need to associate the type of diseases in the hospital, in the patients, when they come to see you. How do you classify them, and how do you diagnose, or how do you identify whether these patients are having infection from the RNA or the DNA viruses? So hopefully towards the end of the session, you will know how to differentiate them. And there are lots of terminology, there are lots of nomenclature. How do we name the viruses based on the properties? So you will learn this too, also. And finally, what is the lab diagnosis of viral infections? And I believe you have gone through your PBR session 1, and some of the viruses might be significant for you to understand further. All right, so if you got any question, just don't be afraid, just raise up your hand and unmute, speak to me. First thing first, you need to define what is virus. We've been talking about this for a couple of years back, since the pandemic, okay, since the SARS-CoV-2 pandemic, and people keep on complaining about these small virus that invaded human being at large. And virus is small living particles, very tiny and minute creatures that can infect cells and change how the cells functions. So some people say virus infected human, yes, but it goes into the human and infect the cells. That is the main aim. And why does this happen? Why the virus need to invade our system? Of course, as I mentioned earlier, it's for their sustained life. Okay, they need to replicate, they need to double themselves, they need to survive. That's why they invade our system. The virion, on the other hand, is a complete viral particle with its DNA or RNA core and the protein code as it exits outside the cells, and also called an infectious viral particle. So virus is on one single organism, but it's constituted or comprised of many components. Okay, they have the subcomponents. Inside the virus, we have the DNA, we have the coatings, we have the proteins, we have the carbohydrates, we have lots of machinery for them to hijack our system. All right, so when we mention about the virus, it's not only one small single piece of creatures, but it's a combination of subcomponents, and we will further look into that too. Viriot is a nucleic acid without the protein coat. What is the protein coat? What is nucleic acid? You have learned about this. We know that the main nucleic acid is DNA or RNA form. DNA is the deoxyribonucleic acid, which appears in the double-stranded, whereas the ribonucleic acid or RNA, they will appear as a single-stranded nucleic acid. And both of these genetic information are inside the virus. Okay, some virus being classified based on this genetic material, either DNA or RNA. It cannot be both. Okay, you can choose only one particular genetic material, not more than that. And virial is small infectious agent of plants. Okay, the virus that infect the plants in the form of nucleic acid without protein coat, we call it the viriot. Okay, like the potato, some of the potatoes that you have eaten, most commonly they have been invaded or infected with this viriot. And if you consume those infected potatoes, you will have some symptoms. All right. Prions is considered as the disease, and is causing agent, and that is neither bacterial nor fungal nor virus and contain no genetic materials. Okay, prions is not a virus, it's a disease. All right, causing agent that is not a bacteria, there's not a fungus, not a virus, and they don't have any nucleic acid. So we will look further into that. So general characteristic of virus. Tonight we want to learn about the viruses. What is the overview? What is the general characteristic? How do you want to know that this is a virus? Okay, that's the virus in the computer, in your computer, in your laptop is considered as the viruses too. All right, so the general characteristic of viruses, it can be classified as obligate intracellular parasite. Some of the virologists, they give this definition. Obligates mean they are compulsory. Intracellular parasites, intra means inside, cellular is the cell, so inside the cells. Parasites, okay, means they invade the system and they try to destroy the whole system or the whole tissue or organs. All right, so these are compulsory things to happen. Viral infection, like a human being, you are infected almost more than 1,000 different types of virus throughout your lifetime. All right, so you have to admit that you are dependent on your immune system. All right, some viruses are lethal, some viruses are not so lethal. All right, so you have to know what are the names of the virus, okay, how they are where they are originated from. General characteristic of virus, the second one, they contain a single type of nucleic acid, as I mentioned earlier, either DNA or RNA. They have the protein coat. Protein coat, it means the covering or the outer layer that sometimes is self-enclosed by an envelope of lipids. Okay, there are some lipids or the fat or lipid covering, okay, that covers the structure of the virus. Okay, we have the carbohydrates, we have proteins. This is only the general characteristic, because some of the viruses, they not necessarily need to have all these structures. Okay, so these covering or protein coats, we call it a capsid. Okay, the capsid. And the virus, they multiply inside living cells by using the synthesizing machinery of the cells. It's very intelligent creatures. Even though it's a very small creatures, the virus can invade the cells and using the energy from your cells in order for them to replicate. Okay, so it's a very intelligent and also a very opportunist living creatures. They can hijack your system and make you sick. Like me, yesterday I just infected by virus, not sure what type of virus, but I'm sure that this is the flu virus. Okay, the flu virus has infected me and causing the symptoms. like the a flu and also a headache. Okay, just a quick example. And the virus causes the synthesis of specialized structure that can transfer the viral nucleic acid to other cells. As I mentioned earlier, they are very intelligent creatures. They can make their own protein or little protein and transfer it to the neighboring cells, and the neighboring cells can die too. All right, so be careful with the virus. Some of them can cause you lots of problems, and towards and also at the most worst cases, they can cause a lethal. Okay, you will die because of the invasion. The host range. So as I mentioned on the slide, you can see that this is one example of the virus. So you have the head on top, whereas they contain the nucleic acid. In the case is DNA. They have the neck, helical sheet on the outer layer. They have the tail fibers. Okay, they have the tail, they have base plate, they have the bacterial surface. So you need to know that the virus is smaller than the bacteria. Okay, the virus is smaller than the bacteria. Hence, they can invade the bacteria and kill the bacteria. They can hijack the immune system of the bacteria and start to reproduce or replicate within the bacteria. Same goes to human. If the bacteria, if the virus invade human cells, they will hijack the system and start to make more copies of this virus. So when they have a good amount of virus inside our system, they will leave the system and invade a next person, for example. But in this case, this is the bacteriophage. The name of the virus is bacteriophage. Why the name is called bacteriophage? Because it's very specific virus that only invade the bacteria. That's why we call it bacteriophage. Means they only phage, they only invade or hijack the bacteria. The bacteriophage doesn't invade the human system, okay? We have the SARS-CoV-2, we have flu virus, influenza virus. So these type of virus invade human, not invading the bacteria, all right? So the host range means how is the range of the, how is the range for the invasion of the bacteria? What types of organism that the virus can invade. So they require very specific attachment to the host cells. Of course, this is important for them to recognize and cling onto the system. And with this strategy, they can modify the DNA or RNA content in the cells and hijack the whole system. Okay, so if you look at the viral size, so at the bottommost, in the rod light, that's the E. coli. You see at the bottommost, that's the E. coli, which is the bacterium within 3,000 times 1,000 nanometer. Yeah, so this is the E. coli's size. On the right hand side, you can see it's human red blood cell, which is 10,000 nanometer in diameter. So it's bigger than the bacteria. But those on the white surface, on the black space in the slide, you can see we have bacteriophage T4, you have the rabies virus, you have chlamydia elementary body. These are all the viruses that are most commonly associated with the human diseases, okay? Also with the plant and bacteria-borne diseases too. So if you look at the structure, there are various structure. You can see the Ebola, like a letter B, is like a club-shaped Ebola virus. Some of them look like blocks. Some are just like a small dot. Some appear with lots of receptors on the outer surface, like adenovirus. Some rabies virus look like the plant stem. So some look like elongated spiral. So there are thousands of different shapes or morphology of this virus. So you need to know what is the basic structure, and how do we classify or how do we name them. So we determined with the aid of the electron microscopy, because it's very tiny, because it's just like 100 to 1000 nanometer. So it's quite difficult to observe by the naked eye. Okay, so the range is from 20 to 100, sorry, 20 to 1000 nanometer in length. So if I ask you in the quiz or in the examination, you must be able to know the size. All right. So the virus structure, the virion is complete, fully developed infectious particles, a composed of nucleic acid and surrounded by the protein coat. Protein coat is the most common structure that functions to coat the virus or the viral component. Okay, but some viruses, they don't need this protein coat. They can survive on their own. Nucleic acid, as I mentioned earlier, some viruses, they have a DNA as their genetic material. Some of them have RNA, but never both. Okay, only either one. It can be single-stranded. So this is the tricky thing. You know that the DNA is the double-stranded nucleic acid. RNA is the single-stranded. But in the case of the virus, the DNA inside the virus can be single-stranded or double-stranded. Okay, it can be linear or circular, like the bacteria DNA. We call it as a plasmid. Plasmid is the bacterial DNA. It appears in a circular form. So in the case of virus, you can find that the DNA or RNA can appear in a linear form, means a very straight line, or circular, where like a loop structure. Okay, this is also possible. And the amount of DNA or RNA varies from a few thousand nucleotides to as many 250,000 nucleotides. So nucleotide is the basis, okay? Nucleotide basis that forms the structure of DNA or RNA. If you remember in the matriculation center where you learned about this, sorry, not the matriculation, the CFS or Center for Foundation Study in biology class, you have been taught about the nucleotide bases that makes up the DNA and RNA, which is the AGCT. So A is one base or one nucleotide base, G, C, okay, T. So they have around 250,000. Okay, that's the maximum numbers. Means the length of the DNA can determine what types of proteins or how big is the protein they're gonna produce. And obviously, the bigger virus, they are producing a longer nucleotide bases. Okay, longer DNA. If you look at the virus structure, we have the capsid and an envelope. The nucleic acid of virus is surrounded by capsid, which is the protein coat, and each capsid is composed of protein subunit called capsomeres. And the arrangement of capsomeres is characteristic of a particular type of virus you call icosahedral or helical. So you have to know this arrangement or orientation, and also the morphology based on the symmetry. Okay, some viruses have capsid covered by an envelope. Some envelopes are covered by spikes, like the SARS-CoV-2 that caused the COVID. They have the spike proteins. You can see they are carbohydrate protein complexes, means that the carbohydrate, they bind to the protein complex that project from the surface of the envelope. Don't worry, because I will show you the slide or the image after this. So this is the common structure that is used by viruses to attach to host cells. Some viruses, they need help from the spike receptors, or that the projects from the outer surface of their structures. Some virus doesn't need this, like the Ebola, they don't need this spike. But adenovirus, they have like these sharp ends, like the needle on the outer side, in order for them to cling and also to bind to that host cells that they want to invade. This is the basic structure, because some viruses, they have envelope some virus, they are non-envelope, means they don't have envelope, as I mentioned earlier, and they don't have capsid. All right. And general morphology, if you look at the structure itself, based on the size, also different. If you look at the morphological types based on the capsid, that is how we name them. Okay, we look at the capsid structure inside the virus, and then we name them. Okay, whether they are helical or polyhedral, icosahedral. So there are lots of these symmetrical surfaces that determines the virologists in order to them for them to assist them to name that viruses. Out there are lot more viruses that is not yet been discovered, and in fact, like the COVID-19 virus, we have like lots of mutations, and that can also change or render their morphology. So again, the virologists can come in and also start to identify or give their name based on this capsid shape. All right. The helical viruses, they are viral nucleic acid is found within the hollow. Okay, sorry, the viral nucleic acid means the DNA or RNA, they can be found in a hollow cylindrical capsid that has a helical structure. Means they store the genetic material inside this hollow structure. We call it a helical structure, and that is the helical viruses. And the most common viruses that have this helical shape is the rabies virus and Ebola virus. The second one is polyhedral. The capsids have the shape of icosahedron. Okay, you have to find what is the icosahedron form. What is icosahedron shape, because they have different multiple symmetrical surfaces. And this is true for adenovirus and poliovirus. So this is how we name them. Some of them we name them based on the size, based on the capsid, based on the lethality, based on diameter, based on where they, what is their host cells. So there are lots of consideration when the virologists want to name a particular name of virus. If you look at again the viral structure, the envelope virus, they are roughly sphericals like influenza virus. I think SARS-CoV-2 also lies in this group. We call it as envelope virus. We have herpes simplex virus, which is HSV. All right, you can see that the influenza virus, the envelope helical virus, whereas HSV is envelope polyhedral virus. Okay, helical and polyhedral is different. Complex viruses, this is the special virus because they're not invading human being, but rather the bacteria. So we call it bacteriophages. That is the one that I mentioned earlier, that they have the head, the tail, the neck, everything like a octopus. All right, give me a few seconds. All right, here you go. Can you see in the slide? I hope you can see the images of the virus. You have A, B, C, D in the figure. The top row is the cartoon, okay, the illustration. But the second row, you can see that's the real images from the electron microscope, and down there I put some of the description for you to see inside the real one and also the illustration. As I mentioned earlier, you can say that the virus ranges from 20 nanometer to 1000 nanometer. And you can see that the helical capsid on the left-hand side, which is tobacco mosaic virus, the capsid is in helical shape. The capsid, okay, you remember the capsid, that used to store the DNA or RNA of the virus. It's very important. Without the DNA or RNA, the virus cannot survive. Okay, they cannot replicate. They will die. So that's why the machinery or the system inside the virus, they have this helical structure in order to secure the DNA material. Like in the human being, if you look at the mammalian cell, we have the nucleus, and inside nucleus we have the what? We have the chromosome. The chromosome is the accumulation of the DNA structure. So in mammalian or in the human, our DNA is stored inside the nucleus. But for viruses, our DNA or RNA, sorry, but for viruses, their DNA or RNA is stored inside the capsid. Look at the B, which is adenovirus. It has an icosahedral shape, a capsid with a glycoprotein spike at each vertex. This is very interesting shape. Okay, so you can see the NA is inside the adenovirus. They have the glycoprotein on the outside. They have capsomeres of the capsid. So the capsomeres is an outer layer that makes up the capsid. Okay, capsomere is the subcomponent of the capsid. You look at the influenza virus, just like the SARS-CoV-2, because they are of the same origin. So it has an outer envelope. Okay, the influenza virus is considered or classified under envelope virus. You can see it started with glycoprotein spikes. The genome consists of 8 different RNA molecules, each wrapped in a helical capsid. Again, you can see inside the illustration, in the inner side of the influenza viruses, they have how many capsid? You have around 8 capsid, and each capsid contains 8 different RNA molecules. Okay, it's very special virus. Okay, and you can see at the image, you can see the bacteriophage T4, like the other T, even phages, has a complex capsid consisting of icosahedral head and tail apparatus. You can see this is like the octopus, as I mentioned earlier, but this is rarely invading human. Rather, they invade the bacteria like E. coli and some other plants too. Okay. All right. So how do we classify? How to summarize it? So we have DNA and also RNA viruses. For the DNA viruses, you can classify them based on the 3 main structure, which is icosahedral, helical, and complex structure. Under icosahedral, you can see is either naked envelope or naked or envelope cytoplasmic. For the naked one, you can see this SS linear. SS means the single-stranded. Okay, DS here is the double-stranded. So SS linear means single-stranded, but is in a line. Okay, just a linear structure. As I mentioned earlier, some DNA, they can form in a straight structure or in a loop structure. Okay, they can form like a circular structure. And they can be both in single strand, only one strand, like a one string, or they can combine or complement with other. We call it double-stranded. Okay, so this is the SS linear, positive or negative. So positive means that they can produce their own RNA. If you look at your lecture notes when you are in the CFS, you know, in the central dogma, when you learn about the biology, okay, in the human sites or in the cell where you have the DNA, right? So the central dogma is DNA will be transcribed into the RNA, and RNA will be translated into the protein. That is the central dogma, which means that your genetic material, which is DNA, they will be converted into RNA, and then the RNA will be converted into the protein of interest. Okay, so for example, if you have any minor cuts that cause you bleeding at your finger, so the body need to produce a new protein, right, in order to seal the injury site. So what happened is that the DNA have this genetic material that encode for specific protein, but DNA is inside the nucleus. DNA need to transcribe into RNA. So DNA is double-stranded inside the nucleus. They need to be converted out from the nucleus in a single form, in the single-stranded. And that's why we call it as the RNA. Some book mention is the messenger RNA or mRNA. Once inside the cytoplasm, the mRNA will be transferred into the ribosomes. The ribosome inside the cytoplasm of a cell, they functions to produce the protein. Okay, so from DNA, they being exported out from the nucleus into the cytoplasm, and then the mRNA will travel into the ribosome for protein synthesis. Now, you have your own protein inside your cytoplasm, and it needs to be transported to the location of interest. For example, if you have a injury site, so this the new proteins that functions for regenerating new tissues or new cell will go there and give or cure the injury site. That's why it's very important for the human being to have this DNA. Means for any diseases, you can the cells can instruct whatever type of protein that is important to cure the disease. Same goes with the DNA viruses. We have the DNA in a positive strand, means they can be immediately transferred into mRNA or RNA for giving the type of proteins or type of action that they want. Okay, so how about the negative one? The negative one means the virus don't have the capability to directly transfer DNA into not transfer, to convert DNA to RNA. Okay, so they need to have an help from the other machinery or other system in order to replicate the DNA. All right, so in this negative SS linear, it means that it cannot be directly converted from DNA to RNA, but they need a special mechanism. They need a help from other RNA to help them to produce the mRNA. So this is a bit complicated. I don't want to confuse you, but as long as we go through this one, I will further explain to you. For the positive one, it can directly produce the RNA, but for the negative one, the virus cannot produce their own RNA. Okay, that is enough for you to know. And for the naked icosahedral, also we have the double-stranded in the circular form. And down here at the blue, in the blue area here, you can see parvovirus. We have papova virus, negative, adeno virus, okay, negative, 70 to 90. So you can see all here are different types of DNA viruses. We have hepatna viruses, but it's a plus sign, which means that they can transfer, they can convert directly their DNA. Herpes is negative. They don't have the capability. They need to make their own RNA. Irido, baculopox. Okay, so these are all the different type of DNA viruses. The most interesting things with the viruses is that they have their own characteristic. And it's important for you to know which structure and what type of disease that they are causing. Okay, so you don't need to know about this DS10 or the genome architecture. It's enough for you to know what type of viruses. Is it DNA or RNA viruses? What is the capsid shape? They are naked envelope, or what is it, combination of shape? So you just need to know about the basic information. Okay, and of course, if you look at here, RNA viruses include a family name like reovirus, Birna virus, Calici, Picorna, flavivirus, Toga virus, Retro, Corona, COVID-19. Okay, here, filovirus, rhabdo, Bunia virus, orthomyxo, paramyxo, and arena viruses. And you can see that the virion diameter is the range of the size, right? So that's the range of the size. As I mentioned, roughly they are between 20 nanometer to 1000 nanometer. So this is the RNA viruses. If someone asks you what is the coronavirus type, so you can straightforward or directly say that it's a type of RNA virus. If I ask you, give me 3 examples of DNA viruses, what would be your answer? You can say it's adenovirus, parvovirus, papovirus. Okay, so I hope you will understand this. So we look specifically for each of these viruses for you to further understand the real structure and also some illustration that has been provided. You can see the helical virus. You have the capsomere on the outside. The capsomere is the small subunit that makes up the capsid. And inside the capsid, we can find the DNA or RNA. So this is the helical viruses, for example, Ebola. So Ebola is classified under helical viruses. Polyhedral, as I mentioned earlier, like adenoviruses, masked adenovirus. So they are also considered as polyhedral viruses. Envelope virus like coronavirus, so influenza virus, they are classified under envelope virus. Inside it, you can find that in the case of influenza virus, they have 8 different capsid in it. Yes, very special one, means they have a special layer to cover apart from the outer layer here. So there are lots of what we call layers. Why the virus, like why the coronavirus needs multiple layers? The simple answer is that they want to cover their DNA or RNA material. They want to protect them, because if there's only one single layer in the virus, so it's easy to be broken up, and also they will easily die. So we don't want this to happen. The virus are very intelligent creatures. They can make multiple layers in order to protect their viral component or the DNA. The bacteriophage classified under complex virus. You can see they have the head, sheath, or the neck. They have the tail fibers, and these are commonly associated with the bacteria. Let us look at the definition. Taxonomy is another branch of knowledge where we are interested in the organization of organisms based on the presumed natural relationship. Okay, classification, you can see this is how do you classify them? Okay, what is the old viruses like a polio, Ebola? They are quite old viruses. What is the modern viruses? They are coronavirus. They are like influenza viruses. Okay, so monkeypox, for a monkeypox viruses. So they are quite modern type. So how do we classify them? Nomenclature is basically how do we name the viruses. The taxonomy of viruses. Before we go, is there any question from the floor? If you want to ask, you are free to do that. Okay, so no question. We go to the taxonomy of viruses. Families of viruses are based on the nucleic acid type, which is DNA or RNA. Strategy, how do they replicate in the host cell? Host cell is the cell that the virus invading. Okay, and then the morphology, the shape, icosahedral, polyhedral, or do they have the capsid or not? What is the structure of the capsid? So we look this already, the structure or the morphology. You look at the genome properties, as I mentioned, single-stranded DNA or double-stranded DNA, the shape of DNA or RNA, whether in the form of sequeler. What else? We have these physico-chemical properties. We have viral protein properties, the genome rearrangement, or the way that they replicate in the host cells. We have the antigenic properties. Some viruses, they have these spike protein or receptors that cause agglutination. Some virus, they can kill immediately or can cause agglutination of the blood. And we also classify them based on the biologic properties like mode of transmission, whether it's from the airborne or from the food or from the touching. So we look at the vectors. Is there any carrier for these viruses? Like some viruses invading the insects and suddenly the insects transferring the virus to human. So the tissue tropism, how do they invade the neighboring cells? So there are lots of properties for these viruses, and hence the name are different. The characteristics are different. Okay, so this is the nomenclature. How do we name them? Of course, it starts with the family name, like the Viridae. So if I tell you what is the family name, okay, so you mentioned like herpes viridae or adenoviridae. Viridae at the back means or referring to the family name. But how about the subfamily? Subfamily like a Viridae, okay? This is the subfamily like beta herpes viridae. And the genus is the virus itself, which is a specific virus like genus herpes simplex virus, but you need to know that the herpes simplex virus also have a different types. Okay, and the different types we call it the virus group like HSV1 or HSV2. Okay, and virus families, we are going to one by one on the virus that we have mentioned earlier. So this is a type of DNA virus. It's a single-stranded DNA non-enveloped viruses, parvoviridae. Some book mention it as the human parvovirus. This is the shape in the form of icosahedral. You can see the viruses invading mostly the kids, the babies. Adenoviridae, viridae at the back is the, as I mentioned earlier, it's the family name. So this is the adenovirus, mass adenovirus, also classified as this double-stranded DNA non-enveloped virus, and they cause the respiratory infections in human. You have the dsDNA or double-stranded DNA non-enveloped virus. For example, papilloma virus that cause human warts diseases, a polyoma virus. The family name is Papoviridae. So I hope you still remember the earlier slide that I have mentioned. Double-stranded DNA enveloped virus, pox viridae. This look like smallpox or cowpox or monkeypox. This is caused by orthopox virus or vaccinia and smallpox viruses. Double-stranded DNA envelope virus, as I mentioned earlier, there are lots of group name or small name under the herpes viridae. They all cause this herpes disease, like the simplex virus HHV1 and HHV2, varicella virus HHV3, so that's another name. Lymphocryptovirus HHV4, cytomegalovirus HHV 5, roseola virus HHV 6 and 7, Kaposi sarcoma is HHV 8. And some herpes virus can be related in the host cell. Some virus, they are very clever. As I mentioned, it can stay in your system for more than 1 year, maybe 2-3 years without giving the effect. You're still alive, but you don't have the diseases. You don't have the symptoms. It's just like the virus are sleeping in your system. But at some other time, they will start to become more active, and then you will immediately get the symptoms. So, okay. So this is the Hepatnaviridae family, most commonly causing the hepatitis. Just in this case, Hepatnaviridae family cause the hepatitis B. The machinery means the system is that they use the reverse transcriptase to produce DNA from mRNA. This is a very intelligent capability of the virus. Okay, you know that this is under envelope virus. This is a DNA virus. So they use a reverse transcriptase, which is a specific enzyme that can reverse. Okay, that can reverse from DNA to, usually it's from DNA converted to mRNA. But in this case, they can produce the DNA from the mRNA. It works backwards. Okay, they have this capability. Some viruses, they are very intelligent because they have this reverse transcriptase enzyme inside their viral component. Okay, so they can convert backwards, okay, from mRNA to DNA. They produce their own DNA, okay, with this enzyme. Some viruses don't have this reverse transcriptase. They cannot reverse, they cannot make their own DNA. All right, so with this Hepatnaviridae family, they have this special enzyme, reverse transcriptase. They can convert mRNA to the DNA. All right, and this is the example of the enlargement of the hepa, means the liver. The female Cambodian patient presented with a distended abdomen due to hepatoma resulting from the hepatitis B viral infection. Some of the viruses, like the RNA viruses that are non-enveloped, like enteroviruses, they can cause this bone malformation, and you can see this is considered as the poliomyelitis or polio disease. You can see the bone of the kid, and also in the eyes, you can see it's become yellowish. Under this category, single-stranded RNA also include the rhinovirus and also hepatitis A virus. For the single-stranded RNA, positive strand non-envelope is the hepatitis E virus, also norovirus that cause gastroenteritis, inflammation of the stomach. Okay. This is some of the example for the single-stranded positive strand envelope virus that includes the ruby virus or alpha viruses. This is transmitted by arthropods, maybe through the food, or maybe because of the environmental risk or environmental factor, because you stay in a less hygienic space. Okay, so it's easier to get the infection. Give me a second. Okay, we will continue. Single-stranded RNA, positive strand, and also envelope. You can find under group family or under family of Flaviviridae. For example, arboviruses and hepatitis C viruses that causes the yellow fever. And they can replicate in arthropods, not in human, but these arthropods are being consumed by humans. Like if you eat these arthropods or maybe some of the animals, they eat these arthropods, and after that, they spread this virus into the animal that you have eaten. Okay, so this is a step-by-step invasion. So you will have this also. So make sure for any food that you are consuming, make sure it's clean and you have heated up or cook it um, uh, nicely, right? This is also to prevent the infections. Some virus, they are heat labile, means they are sensitive to the heat change or the temperature change. So if you're increasing the boiling temperature in the water, for example, you can kill the bacteria, and also for the viruses, also possible for them to, I mean, for their coat to be broken easily, all right? Single-stranded RNA, positive strand envelope, coronavirus, coronaviridae family, and they commonly invading the upper respiratory infections. I'm not sure whether I've been infected with coronaviridae, but that's the only virus that I can associate with. So maybe I'm infected with this coronaviridae family that caused me to have a runny nose and this ability to breathe efficiently. Okay. So you have the single-stranded RNA negative strand and its envelope, vesiculovirus. This is very unique one. Lissa virus, okay. Rabies virus cause numerous animal-borne disease. Okay, you can see these viruses. They are very specific in their activity. Like this virus, they can invade the central nervous system, cause you to have epileptic seizures, or you can die immediately. You can have this lot of what we call jerking motion. All right. So some virus, they are not attacking the CNS or central nervous system. They are attacking the upper respiratory tract. Some viruses, they are evading the blood system. Some viruses, they invading the immune system. Some viruses, they invading your liver system. Okay, so the virus, they have a specific mechanism of action in order to elicit the effect, right? It depends on what type of the virus. Like in this case, rabies virus from the wild dogs, cats, or wolf, any wild animals. Okay, they used to stay in their bodies, in these animals' body or in these animal cells, and at some point, if you got bitten by these wild animals, it is higher chance for you to also being invaded or infected with this vesicular Lissa virus or rabies virus. Okay. A single-stranded RNA, which is negative strand, but it is envelope virus. I'll give you some example like a filovirus, Ebola, Marburg virus. So this is a patient in Africa. You can see this bleeding gum with the sign of hemorrhagic diathesis. It can also lead to lethality. Some people, I think the Federal Drug Administration, if I'm not mistaken, the FDA already released the vaccines for this virus or antiviral agent in order to stop the spread of these Ebola viruses. One of the most lethal virus that kills lots of people in Africa. Paramyxoviridae is a bit tricky, a very simple one, but if you look at the structure, they have like some receptors on the outer side, some protein structures on the outer sides, like a peeling or like a needle shape string. Okay, this is the single-stranded RNA, strand envelope paramyxovirus that cause the para-influenza, mumps, and Newcastle disease. And if you look at the cartoon or the illustration at the bottom, you can see the duck or the avian virus. It's originating from the gray one and it invading the pig. All right, and from the human virus that have this virus in blue, also transferring this virus to the pig. And sometimes this human, they consume the pig and also at the same time consume the virus. In some other occasion, the pig can also release this avian. Avian means the livestock, or not livestock, but the like the birds, like the ducks, like the chicken, or sorry, like the hen or the roosters. All these flying animals, okay, they are higher tendency to spread this virus. And again, this virus is circulating around the ecosystem and start to invade human again. All right, so this is a multiple step, because there is possibilities that the virus don't like this avian and then they just invade the mammalian, and after that they want to invade human. This is also possibilities. All right, they can invade yourself directly. There is no problem at all. Okay, for the virus, these very intelligent creatures, they can do whatever they want, but depends on how they comprehend the machinery inside the host cell. If they think that they can invade the system and hijack it, then they will produce a new protein and start to produce more viral component, or else they will die. Because they are intelligent, they will not go to the host cell that have very high immune system, or else they will just kill themselves. All right, they are very selective in choosing what type of host cell that they want to invade. So this is Delta viridae. Examples like hepatitis D virus, that depends on the co-infection with hepatna virus. This is also intelligent virus, because this type of virus, they can have friends. Okay, they can bring friends in order to give the effect or the disease. Multiple RNA strands. Okay, not only single, not only double, but multiple RNA strand, like the influenza virus, whether it's A or B or C. They have these spikes on the outer layers. They can agglutinate the red blood cells, or RBC. So this is quite a little virus. If you got them, but please be careful, and please seek immediate medical attention if you think that you have this fever, lethargic, nasopharyngeal, runny nose, coughing, okay, or diarrhea, gastric, all the symptoms that I have mentioned here. Multiple RNA strand, again, negative strand, Bunya and Hanta virus, they cause the hemorrhagic fevers. This is also lethal. Arenavirus from family of Arenaviridae. They are helical capsid in it, and they are RNA containing granules. They are lymphocytic choriomeningitis, causing the hemorrhagic fever and Lassa fever. Two RNA strands that produce DNA, like lentivirus, virus that cause the cancer. They have this again, the enzymes called the reverse transcriptase to produce DNA from the RNA. Okay, from the RNA inside their structure. Okay, Retroviridae most commonly cause the mutation in your DNA and leads to the cancers. Okay, so hence we call it as the oncogenic viruses, because they favor oncogenesis. Reoviridae, this is common, as I mentioned, some of them using the insects, like in this case, the Colorado tick fever, the Colorado tick, which is the insect that carry this Rio virus. So once the tick bite you, they also transfer the Rio virus in yourself and cause the respiratory enteric orphans. Okay, and also a condition as I put inside the figure. All right, so we have already 1 hour pass, and this is to summarize. Okay, this is to summarize all the DNA viruses and RNA virus that we have learned. And if I ask you in the examination or in the quiz, please name these, whether this is DNA or RNA viruses. So you have to know, you have to use this what we call acronym in order to know what are the name of the virus and how do we classify them. Okay, so DNA virus again includes the herpes virus, hepatna, adenopapova, parvo, and pox virus. And most of the DNA virus are double-stranded. Okay, they are mostly icosahedral symmetry and they replicate in the nucleus, except, okay, there's exception, and this is a very tricky bit because I might confusing you inside the examination. So parvoviridae that has single strand of DNA, okay, don't confuse with the RNA. It's still DNA but in a single stranded form. Poxviridae that has a complex DNA does not have icosahedral symmetry. Okay, this is 2 exceptions which is important for you to know. They have these 3 envelopes. Okay, or out of these DNA viruses, again, 3 of them have envelopes. Okay, which is herpes, hepatna, and pox virus. If I ask you in the examination, what are the DNA virus that have envelopes? So your answer will be herpes, hepatna, and pox virus. How about the naked one? It's the papova, adeno, and parvovirus. Okay, enough with the DNA viruses. Now, the more viruses essentially comes from the RNA virus group. So that include toga, corona, retropicorna, calici, rio, orthomyxo, paramyxo, rhabdobonia, arena, and filoviruses. And most of them, since they are RNA viruses, they have a single-stranded RNA. They are enveloped. Some of them are showing helical capsid symmetry and replicate in the cytoplasm. All right, as I mentioned earlier, DNA is inside the nucleus, right, in the mammalian cells, and in the virus also, they replicate in the nucleus. But in the case of RNA, it's in the cytoplasm. So don't get yourself confused. RNA is inside the cytoplasm. DNA is inside the nucleus. The virus don't have, okay, you need to know this, the virus don't have the nucleus. Okay, they have the capsid. But in the DNA virus, if you look at point number 6, pox virus, okay, underneath you can see it's showing the replicate in the nucleus, right? Replicate in nucleus means that once the DNA virus come in contact with your cells, okay, they will deliver their DNA, okay, and this DNA will go into your nucleus inside your cells. Okay, that's what does it mean, not replicate in their nucleus. Okay, the virus don't have their own nucleus. It means that this is the nucleus of the host cells. Okay, and exception here, you have the Reoviridae, which are double-stranded. It's RNA, supposed to be single-stranded, but in this case, Reoviridae, they are double-stranded. All right, so I might test you in your examination too. What are the exceptions for RNA virus? So you might want to answer that. And 3 of these RNA virus, they are non-enveloped, which are Picorna, Calici, and Reoviridae. 5 of them have icosahedral symmetry, and 2 undergo replication in the nucleus, like retro and orthomyxo. The rest, okay, the other 10, they undergo replication in the cytoplasm. All right, so this is only a similar image that I have shown you earlier. So if you want to look at it, when I put it in the orange color, it doesn't mean that it's canceled. It just want to highlight the important virus. Okay, all right, everyone, you want to continue, or is it enough up to now, or a bit more? You tell me. Someone, are you already sleeping? You fell asleep already? Anyone want to say anything? Do you understand? Is it clear, not clear?
So far, so good, Doctor.
Clear.
Okay, thank you, Shahira Osman. Clear. Okay, so we will continue another half an hour. Is that okay with you guys?
Okay, Doctor.
Okay, but if you fall asleep, just let it be, okay? Don't force yourself to understand these viruses. Growing a virus maybe is a bit ambitious for us to discuss this, but just to illustrate that we can grow our own virus in the lab. But of course, you need to wear a proper PPE in order to handle the virus. Yes, of course, for research purposes, we can use the embryonated egg in order to grow the virus in a large amount. For example, in the production of vaccines. So the vaccines is the weakened virus. So you want to produce more and more of that virus. And after that, you attenuated them or you make them weak in order to supply to your body. Later on, your body will produce antibody against that weak viruses. Okay, so this is just to illustrate how we can grow the virus in our lab. Viral multiplication. This is also the next stage, which is also quite important for SS questions. I think in your examination might be some of them important for the for the multiple choice. So for virus to multiply, it must invade the host cell, as I mentioned earlier, and take over the host metabolic machinery. So you have your own cells. Like me, I already been invaded by the virus. So I considering myself as the host cells. The virus come and invade my machinery. The virus come and use my energy inside my cells in the form of ATP, right? Your mitochondria in your cells producing ATP for your cellular function. The good thing with the virus is that, not a good thing, but it's a bad thing actually. So the virus come and invade your cells. They're using the energy that your cells are producing. Okay, so they're gaining more energy to produce more virus. At the same time, you become weaker. Okay, because you don't have any more energy to sustain your cellular activity. So multiplication of the bacteriophage can be classified into two main mechanisms. This is extremely important. If we talk about the viruses, how do they multiply? Just these two main cycles are involved. Okay, this is only two main mechanisms that the virus can survive. Excuse me. So the first one is the lytic cycle. It ends with lysis and death of the host cell. And this is lytic or lysis, means they are destroying the whole structure. They kill it. So the virus come to the cell of interest or the host cell. They multiply in it. When they have a good amount, maybe 1,000 more than that, or millions of virus, they will destroy that host cell. That is the lytic cycle in general, okay? However, the lysogenic is that they will come and invade, but then the host cell will remain alive. This is only a temporary storage or place for them to live and multiply further without killing the host cell. Okay, so this is two main mechanisms. The virus can choose either one, either lytic or lysogenic cycle. Remember, lysis means they will lyse the cells, the host cells will die and break, but lysogenic, they just come, okay, multiplying it. When they are finished with the job, they will go back, okay, and invade another system. Basically, that is the way. And of course, for each cycle, as we mentioned about the cycle, means they must go through a few steps. So what is the step? The virus will come and attach. They have these tail fibers, like the case of complex virus, like bacteriophage T4. If you mention the virus with like an octopus shape, they will invade the system. They have these small foot, we call it the tail fibers. Okay, they attach to the host cells, they penetrate, they open it up, okay, and then they will inject their DNA inside the host cell, and these cells or the DNA that has been injected is considered as the viral DNA. And with this small viral DNA component, the virus is able to become a mature virus in it. With only one piece of DNA, it can make a new virus. All right, so they don't invade and make their own copy inside. No, they just give their piece of DNA inside. Okay, they just inject their DNA pieces, and with this DNA, you know that DNA functions to produce the RNA and RNA to protein, right? So DNA, they can produce the protein which is considered as the viral protein, and they make a new whole set of a virus copy, and then they become maturation. Okay, they become bigger and mature, and they are ready to release. Okay, so this is an example. It start to attach the bacterial cell wall. You can see the capsid and DNA that they have. Okay, the first step is attachment. Phage attached to the host cells. After that, they penetrate. They penetrate the host cells and they inject its DNA only. All right, so from this DNA, they start to biosynthetically produce a new component of the virus. How clever it is, right? So it's not invading the whole system, because it's quite big for the virus to enter the bacterial cells. So instead, they just give the DNA component. And after that, there are lots of information from the DNA, because DNA can produce capsid, the DNA can produce tail fibers, all the important components of the bacteriophage. And then they become mature, means they have become assembled into whole new set functional virus. Okay, so they are assembled into virion, and when they are ready, with a good amount, they will be released from the host cells and start to invade another system. Okay, so this is called as the lysis cycle. However, for the lysogenic cycle, you can see here the process is quite similar. All right, the phage attached to the host cells and injects this DNA. The bacterial chromosome is in circular shape, as I mentioned earlier. Some books, they mention bacterial chromosome as bacterial DNA, or some books also mention it as the plasmid. Okay, you can name it. All right, so the bacterial chromosome or the bacterial DNA now, okay, in a circular shape, but then the phage attached to host cell and injects DNA in a single stranded, or in the positive strand. That someone is joining us. Welcome everyone. If you miss the line, okay, you can just click on the YouTube later on. And after that, you can see on the left hand side, right? So that left hand side in red is the lytic cycle that will break up all the host cells. But the cells start to go to the lysogenic. This is another cycle. You can see in the middle here, I'm not sure if you can see my arrow here. The virus can choose whether they want to destroy the host cells, or they can just make the host cell as the place for them to multiply. All right, for lethal, basically for lethal virus, they just want to destroy the whole system. For some good viruses, they are maintaining this cellular structure of the host cell. Okay, so you look at number 1, and after that you look at the number 2. Phage DNA circularized and enters lytic cycle or lysogenic cycle. They will decide. And then 3B, okay, you can see that the phage DNA integrates with the bacterial chromosome. This is also a clever way to hijack the system. The viral DNA integrates with the bacterial DNA or the host DNA by recombination. This is very amazing, right? Because they can just, bind whatever location that they want. And what happens is that this bacteria will start to split into two based on the cellular division, and they will carry this viral DNA inside their bacterial DNA. So you got two type of DNA now, because they are combination of virus and bacteria DNA. What happened is that occasionally the prophage, so we call this combination, okay, we call this viral DNA from the virus that is inserted into the bacterial DNA as the prophage. Okay, so the prophage may be excised from the bacterial chromosome by another recombination event initiating a lytic cycle. Okay, so this is a lysogenic way. At some point, the virus that integrates their viral DNA can be excluded or can be cut from the main prophage. Okay, so there might be some bacteria with mixture of DNA. Some bacteria maybe come with the full virus DNA. The other bacteria might be full healthy bacterial DNA. Okay, so there are three different combinations of bacteria that can survive out of this cycle. All right, so basically for those bacteria that retain these viral DNA, they will follow the lytic cycle, they will die immediately. But those who have the combination of viral plus the bacterial DNA, they will still survive. The other bacteria that have only the bacterial DNA will also survive. Okay, so this is one way that the bacteria can survive, sorry, the virus can invade the system without compromising the activity of the current or healthy bacteria that they invade. Okay, so this is the explanation for the lysogenic cycle. They have these pathways or the cycle step by step. I think you can read it through too. The specialized transduction is also similar. Just to give you an overview that this is an example in the case of galactose positive. I would like you to focus on this because it's important for you to understand the whole process. Okay, there are the specialized transduction. What does it mean is that the virus can join or can mix the virus DNA with the bacterial DNA. This is like the lysogenic that we have mentioned earlier. But we have the GAL gene. GAL gene is the name of a genetic portion inside the DNA that encodes for galactose, that give rise to the galactose or the sugar. Okay, so you look at number one on top here, we have the prophage, which is the viral DNA. You have the GAL gene inside the bacterial DNA, and you have the whole bacterial DNA. Okay, the GAL gene is a small portion of the bacterial DNA. Prophage exists in galactose using host containing the GAL gene. All right, so now this is combination of the virus and a bacterial DNA. Number two, fast genome excises, carrying with it adjacent GAL gene from the host. Okay, fast genome excises. Can you see here the fast genome in pink? Okay, they've been cut out. All right, leaving the bacterial DNA alone. All right, but the GAL gene is being carried with the viral DNA. Okay, the GAL gene, the galactose gene. Okay, this is very important gene for the virus because the virus have a very selective capability to choose what is the gene that they want to mutate or they want to invade or they want to choose. Right, so the virus choose only this specific one GAL gene or galactose gene, and then after that the phage matures and cell lyses, releasing phage carrying the gal gene. Because the virus only want the gal gene, so they just cut the gal gene from the bacterial DNA and carry it with them. All right. So number 4, phage infects a cell that cannot utilize galactose or lacking a gal gene. Now, the next activity is that the virus that have the gal gene plus their own viral DNA, they go and infect the bacteria that don't have the gal gene or lacking gal gene. What happens is that along with the prophage, the bacterial gal gene becomes integrated into the new DNA. Okay, you can see the gal gene inside the virus here is become inserted inside a new bacteria that don't have the gal gene. And then this gal gene integrates with that new bacteria DNA. Okay, and the lysogenic cell can now metabolize the galactose. Okay, so that from the galactose negative, okay, bacteria that don't have the galactose gene, they will have this galactose gene. They have the gal gene because the virus carry it from the one that they have invaded, the first bacteria that they have invaded. Okay, so lysogenic cell can now metabolize galactose. So the cell now, the bacteria cell now, instead of dying because they don't have the gal gene, they can now survive. Okay, they can now survive and live happily. This is how is the trick. The virus want to cheat or want to confuse the bacteria. Okay, they try to what we call to cheat or to lie the system, the bacteria system, saying that, okay, don't worry, we don't do anything dangerous for you, you just carry out with your normal activity. But actually they are invading the system. All right, because the bacteria don't like the virus. The bacteria have also immune system in order to kick off the virus. They can just kill the viruses. But the viruses are more intelligent than the bacteria. So the virus are manipulating or modifying their capability to modify the DNA sequences to what do we call, not to cheat or to confuse the bacterial system. I have the question here, is this how the mutation started, doctor? So the mutation is like most viruses, because they are very small and they carry the genetic material. Of course, they can cause DNA changes, whether 1 bases, 2 bases in your system, and then they will cause this oncogenesis, or cancer will easily form. Okay, this is how the mutation started, not necessarily through this way. This is only one of the example. Okay, the virus can directly cause mutation because they have the DNA, right? I told you earlier, the virus have DNA and also RNA. So once they invade your system, they also changing your DNA and also RNA. So you are get higher possibility to get the mutation. But you need to know that the mutations, once you have been invaded by the virus, is not necessarily lethal. Some virus, they can only cause a minor mutation, but right after, your body have a special machinery to repair it on their own. Okay, so you will get a healthy cells again, and that mutated cells will become degraded and being excluded from the system. Okay, they will be removed from the system through your cellular machinery or through the vesicle. All right, so any virus can cause a direct effect on the mutation. This is a more detailed or more advanced version of mutation or we call it as a specialized transduction. Multiplication of animal viruses, you can look at this. This is also quite similar. We look at the steps that is involved in regulating the activity or multiplication of the viruses inside the body. Okay, so this is a multiplication of the virus. In the case of parvovirus, you can see this is a type of DNA virus. They attach to the host cells, they penetrate, they go to the nucleus, all right, and then they start to multiply, do the transcription from DNA to RNA, and after that they produce the proteins, and this capsid protein can further be released as a virions, okay, a virion, a mature viruses. This is how basically for the parvovirus. Not all like this, but generally this is the way for DNA virus to multiply themselves. I put some of the biosynthesis of RNA. How do we make them, and what is the special features in their biosynthesis, in the making of the new viral copy? Okay, new virus copy is different because some viruses they need to produce the capsid or the protein code. Some of them don't need to produce the protein code. Okay, so here I put like here RNA positive strand like Picorna viridae and Toga viridae. They have this capability to synthesize RNA polymerase, which copy negative strand RNA to make mRNA in cytoplasm. Okay, so this is how we make the virus. As I mentioned earlier, if you have this DNA from the virus, you can make your own virus in the lab. Just make sure you have this DNA sequence that code for what type of protein. Okay, let's say structure A, DNA for capsid. Second one is for the protein code. Okay, the third one is for the tail. Okay, or for the helical capsomeres. Okay, if you have this complete combination, then you can just combine them all together, and then it will become a complete functional virus. All right, so this is the way how do they multiply for RNA, not for DNA. Okay, this is quite busy slides, but I want you to focus on the DNA instead of RNA, because you might be confused with the ssRNA negative or ssRNA positive. I don't want you to get confused. So go for the easier one, which is the DNA. Here, I think we're going to the last part of the lecture for today, viruses and the cancer. Most of the cancers are originated from the viruses, but we cannot exclude it that the cancers also can come from the genetic, hereditary, from your mom and dad. It can be from the environment, excessive exposure to the chemicals or to the ecosystem, or from environmental exposure like the pollutions. This is also factors that cause the cancer apart from the viruses. But today I'll just want to summarize that the virus is one of the main contributor to the cancer. All right, and we call these viruses as oncoviruses. They are the viruses that capable of inducing tumor or causing cancers in human or animal. All right, and I list down what are the viruses, DNA or RNA viruses that is considered as oncogenic viruses, and the loss of diseases that has been associated with the virus, like Epstein-Barr virus causing the Burkitt's lymphoma, has hepatitis B virus that causing liver carcinoma. Liver CAS is liver carcinoma. Okay, so this is one of the example. You have the throat cancer, right? This is also caused by the viruses. RNA viruses or RNA oncogenic viruses, you can see here. Virus, latent viral infection. Okay, a virus can remain in equilibrium with the host and not actually produce disease for a long period. I already mentioned about this. They can just sleep in your system without you noticing that. And they most remain in the nerve and wait for decreased host resistance, then perform a sudden attack. Okay, it's like a bomb, okay, like a time bomb. They can live with you for more than 10 years in your system without you realizing. Suddenly you get this sudden attack. And this is true for the case of herpes virus like HSV, chickenpox virus, varicella zoster virus that cause shingles in the kids. All right. Okay, this is shingles. Okay, and just now is the latent. This one is the persistent. A persistent infection is the one that occur gradually over a long period of time. You have these symptoms or process occur quite chronically. Okay, every time you will have this. Measles virus, for example, causing encephalitis, like the subacute sclerosing panencephalitis. Okay, this is example. If I ask you, give me example of the latent and also persistent viral infection. Okay, so we will give you you give me the answers. The prions is the disease. It's the based on is the acronym for the proteinaceous infectious particles. And the disease under prions are called as the mad cow disease, Creutzfeldt-Jakob, Guttmacher-Straussler-Scheinker, and fatal familial insomnia. This is all the prions. It can cause by the genetic factor or maybe from the feeding scrapie, which is the neurological disease in the sheep, infected sheep meat to cattle and eating undercooked beef from infected cattle. So if you have cow, slaughtered cow, goats or sheep, calf, okay, so these livestock or these animals where you used to eat their meat or lamb, for example, most commonly they are infected with the viruses. Okay, so make sure you have to clean it up properly and also cook them properly cooked, okay, not undercooked. And the Creutzfeldt-Jakob disease, CJD, is commonly a disease that implicating in the nerve tissue complication. Okay, so you will have this central nervous system problems. Apart from that, the plant viruses also can cause lots of complications to the plants. Viriots, potato, they are also infected by the viruses. Not only animals or human, the plant also come into this category. Okay, so I put all the examples, further examples, and some of the diseases. I hope you can read through. Because you need to associate. If I ask you what is the disease that is caused by the Epstein-Barr virus, so you will answer it's the Burkitt's lymphoma or infectious mononucleosis. Okay, what is the condition that caused by the hepatitis B virus? So you will say that, okay, it can be hepatic cirrhosis. It can cause hepatitis B. All right. So you need to associate between the type of virus, the name of the virus, and the disease that they cause. Okay, the disease that they are causing. CMV, cytomegalovirus. One is the disease that they are causing. So you can see it's causing the mental retardation. Okay. So a latent infected cells swell, okay, swelling of the infected cells, cytomegalovirus infections. Okay, some of this is just further explanation for you to go through and read through, because I have added lots of example here. Previously we have a look at the structures, and here I put the name of the virus plus the disease that they are causing. Okay, plus some of the minor information that I think is beneficial for you to know, but try not to remember all things. Okay, the most important is to understand what is the structure, how do we classify them, based on what. Okay, so I take example here, like the coronavirus. As I mentioned earlier, they are quite unique because they are quite similar between each other, like influenza virus and also SARS-CoV-2 virus that caused the COVID-19. Roughly, you can see the structure is quite similar, but of course, there are many differences. If you look at the influenza virus, there are 4 strains and multiple subtypes. They are negative strand, they are segmented, they have the HA and NA surface protein. Hemagglutinin, I think, and NA is neuraminidase, if I'm not mistaken, is a type of protein at the outer layer. But the SARS-CoV-2, uniquely, they have this spike protein, and that is the main differences. And both of them are coronavirus, which are under envelope virus. Okay, you look at the flu and also the COVID-19. What are the differences? For COVID-19, they have additional symptoms like loss of taste and smell, red and swollen eyes, and skin rashes. Okay, measles virus, mumps virus. Okay, measles, rubella, measles virus. Can you see that causing encephalitis? Okay, that's why you need to go for the vaccination, but vaccination is not can be said as the 100% the best cure. Okay, vaccination is not the only answers. There are lots of other possible answers to cure this viral infection. Okay, so please do not restrict yourself. When we mention about the COVID-19, you have to immediately get the vaccination, but rather please seek your general practitioner or the doctors for advices. Why I'm saying this? Because for taking the vaccination, you need yourself being screened properly. If you have any immune diseases, if you are immunocompromised, okay, so you are not, your body is not suitable to receive any vaccines. In other words, your body cannot tolerate the vaccines. So make sure, please, please, please consult the medical authority or any medical doctors. What is the best solution? Okay, we cannot vaccine people, immediately. It needs to be on the basis of the need. Okay, if you need it and you can tolerate it, and go for it. If you don't need it because you have any other complications or diseases, try to consult your doctor. Okay, rubella, macular rash, fever. Okay, prevented. Most of the virus, we have the antiviral that is already FDA approved. Some of them, some of the vaccines are still under manufacturing. Some viruses are a bit outdated, but recently, like the polio, it comes again after a few years. I think after 100 years it comes again, or maybe 50 years back. Okay, so this is just a very rare viral. And this is the HIV. How do we detect it in the hospital? Hepatitis C virus. What is the lab diagnostic? Rabies virus causing the rabies encephalitis, mode of transmission because you got bitten by the infected animals. And what happened is that specifically the rabies causing the disturbance to the CNS, okay, the central nervous system. So immediately you need to go to the hospital if you got bitten by these wild dogs, or the wild monkeys or wild animals. All right. For this rabies virus, they have a few stages. 4 main stage, we call it as the prodrome clinical stage. The first stage, you have the fever, headache, fatigue, painful a sensation at the bite side, and after that is acute encephalitis. Your brain, you have the brain fog of confusion, seizures, and after that, brainstem encephalitis, frothing of the mind, and it can cause the death too. So make sure quickly, once you get bitten, don't allow the virus to multiply inside your cells. Rather, quickly go to the hospital. Okay. Most commonly, rabies virus we treat with the immunoglobulin or antibodies or just vaccines. Right. This is an example, the step when the virus enters the tissue from saliva, and after that, it can cause the death. Okay. So the time is the only savior for you at this time point. So this is only population of the percentage. What is the carrier of this rabies virus? You have dogs, cats, rodents, rabbits, coyotes, fox, bats, skunks, and raccoons. They are all the carriers for these rhabdoviruses or rabies virus. All right. So make sure you take good care of yourself. And the polio too, same thing. For this part, I put some of the associated diseases and how did they got transmitted to your system. All right. So I think it's enough for tonight, because you can just read the rest of this slide. It's not that different from the one that I have mentioned earlier. All right. So in the lab, if you want to confirm if this patient has been infected with a certain type of virus, what is the lab test that we most commonly do? We use the tissue culture. We can use the fluorescence antibody staining to check what type of antibody that has been released in order to prevent this viral infection, maybe IgG or IgE or IgM, you name it. Okay. So ELISA is another type of assay that we can test or to check to confirm that the patient is having the viral infection. Some other method like nucleic acid probe, it can generate or make their own nucleic acid and complement it to the viral DNA or the viral nucleic acid. This is for the detection purpose. If the virus have AGCT, so you will make complementary AGTC, okay, to that viral, so that you can detect and confirm whether this is a viral infection or not. Otherwise, you can test the confirm by using the PCR, polymerase chain reaction, where they multiply the DNA of the virus many times. Okay, from one copy can go to millions of copies, so you can know that this is from that particular virus. Serology, using the antigen and antibody reaction, you can see the increase in the immunoglobulin M, or 4 times increase in the immunoglobulin G antibodies. You can see if there is rise in the IgG in your body system. So that is one also one of the method that we can use in order to detect or confirm that your body is now under viral infections. All right. So I hope that's the end of our session today. If you want to have the copy for this section, for this session, I already recorded and live stream it in the YouTube. So you go to my YouTube channel and you can just quickly grab some important points for today's lecture. I will spend around 5 minutes for you. Please, if you can use your own real name, because we're going to have a short quiz for tonight. So we have around 81 people on the line. But before that, let me answer one question from Hazik asking, CJD only happen to patient with some sort of surgical tissue problem, right? So people who never had surgery would never get CJD. CJD, yes, CJD, when you got infected with the utensils or the apparatus, the surgical apparatus during the surgery that is already infected by the virus. But this is not 100% only the only way that the virus can infect you. There are lots of other ways or other mode of transmission. Okay, by contact. Okay, direct contact with the person who have this CJD also is possible for you to get the same disease. All right. So you look at back at the notes and find out what is the mode of transmission. Okay, so I hope you are still on the line. We have the quiz now. So please quickly within each question is only around 60 seconds. Okay, each question only 60 seconds. So you will answer immediately. All right, get ready on the screen, everyone.
Here, Doktor. Doktor letak link dekat mana, Doktor?
I will pull it up. I will make it as a pool. Okay, get ready. I want 82 answers. Look at your screen in the Zoom. Okay, you have 60 minutes for each. Okay, 1, 2, 3. A virus that enters a bacterial cell is known as. Okay, quickly, which answer? Quickly, we're waiting for the answers now. Uh, 30 more people to answer. We have another 20 seconds. Another 4 people. Okay, very good. So we have all correct except one person. Okay, the answer is bacteriophage, and one person answer plasmid. Okay, all right. Let's move on to the next question. Okay, a virus can be classified by its type of. Okay, you answer it. It's either mitochondria, nucleic acid, chromosome, or carbohydrate. Another 40 people to answer that. We have another 30 seconds. We have 5 more people to answer. There's another 4 people. Please answer the question. Okay, perfect. So yes, another 4 people. Can you answer? Okay, the answer is nucleic acid. Okay, some of you answer it's a mitochondria, some of you answer chromosome. Okay, the answer is nucleic acid. Let's move on to the third question. Which viral life cycle allows viral genetic material to lay dormant while the host cell reproduce? Which viral life cycles allow viral genetic material to lay dormant? I mean, they will allow the host cell to retain or remain alive. Now, which cycle? Is it lytic, lysogenic, mitosis or S phase? Okay, another 20 seconds to go. Okay, so the answer is lysogenate. Okay, the host cell will remain alive. But some of your friends, 7 people answer is lytic. Okay, so it's not correct. It's wrong. Okay, not lytic. It's lysogenate. Lytic will kill all the host cells. All right, so we move to the 4th question. Most RNA viruses are single-stranded, envelope, show helical capsid symmetry, and replicate in the cytoplasm. Except for, okay, remember the slide I mentioned earlier? What is the exception? You got 40 seconds. Another 20 seconds. Waiting for another 30 people to answer. Okay, so I got lots of answers here. It's a mix of answers. So the answer is what? Make sure you answer, okay? Because your answers will be collected. So the answer is orthomyxovirus that undergo replication in the nucleus. All right. Move on to the next question. The last question. Number 5, which of the following differentiates influenza from the SARS-CoV-2 virus? What is it? Which category or which items that differentiate? What are the difference between the influenza and also the SARS-CoV-2? You have 60 seconds from now.
Aduh.
What happened? Is it difficult? This is the last question. Okay, you got 20 seconds more. Another 30 people to answer. Please answer all the questions, because if you don't answer the question, you get 0 for that question. So we only have 5 questions. All right, another 5 people to answer. It's okay if you don't get any question. If you miss any question, it's okay. All right. So very good. I'm gonna end the poll now. Let's see the result for question number 1. Can you see that? Yes, the answer is bacteriophage. Okay, for question 1. Question 2, the answer is nucleic acid, which is correct. All right. Question 3, the answer is lysogenic, because it not kill all the host cells. Question 4, the answer is orthomyxovirus that undergo replication in the nucleus. Okay. Question 5 is A. Okay, the first one. Unlike SARS-CoV-2, influenza virus consists of 4 strains. All right. So I will collect all the answers and try to keep it for my collection, and we'll see if we can use that for your carry marks. Do you want it to be counted in your carry marks? Yes?
No.
No. Okay. It's okay. We'll see. I will just keep it for now. Thank you very much, everyone. Is there any final question before we end the session?
No, sir. No, Prof.
If you got
No,
any
sir.
issues, or you don't like me, or you hate me, so just let me know. Other than that, I wish you all the best. For my class, we will have for biochemistry, I will teach you the hemoglobin synthesis, and also genetic 1 and genetics 2. Okay, so if you got any issues, just let me know. I will try to replace the hemoglobin synthesis class for biochemistry later, because Dr. Samso is going to take the session for week 5, if I'm not mistaken. So we will do the class online later, okay? Because Dr. Samso want to take the session. I think that's it from my part. Congratulations, everyone. Thank you for your cooperation, and stay on the line till late at night like this. I hope Allah will shower His bounty on you and give you blessing. For everything that I have mistakenly did during the class, I will feel very sorry for that. If you don't understand any of the lectures, please let me know. Okay, either through your class rep or just directly to me. Okay, don't feel shy for this knowledge, because it's also important for your subject. Okay, later on for your examinations too. If you don't have any more question, I will just say good night, everyone. Wish you all the best. Wake up early tomorrow for Subuh prayer, and we end the session by Tasbih Kafara and Surah Al-Asr. Subhanakallahumma wa bihamdika ashhadu an la ilaha illa anta astaghfiruka wa atubu ilayk. Wal 'asr innal insana lafi khusr illa alladhina amanu wa 'amilus salihat wa tawasaw bil haqqi wa tawasaw bis sabr. Thank you, everyone. I'll see you again next time.